Today we will discuss a very important topic, which is the origin of microbiology. This topic is actually a very lengthy topic. I will try to discuss this topic in a simplified manner. Most of the students find it very difficult, many students find it very difficult during their exam to actually make a note in this topic, but I made it very short. You can use this for your exam purpose. So let's begin. Before the invention of microscope, people believed in spontaneous generation. They have no idea about germs, the microbes, or any invisible substance. They believe that the organic substances which are present in the food cause disease, and also they believed that diseases are actually caused by some supernatural existence, some evil, or by some gods, curses of some gods. They have no idea about the germ theory of disease or any modern concept. They have no idea. But after the invention of microscope, scientists have finally disapproved this spontaneous generation, and also the development of many new techniques have helped scientists in understanding the ecology and behavior of microbes. Some of the techniques are genetic techniques, genome sequencing methods. Okay. Roman philosopher Lucretius and the physician Girolamo Fracastoro, they found some invisible living creatures that can cause disease. They have no idea that these invisible living creatures are microorganisms or microbes. They believed that there is some invisible living creatures which cause disease. Next, scientist Francisco Stelliuti, he observed bees using a microscope, and that microscope was provided by Galileo at that time. You heard about Galileo. So next, Robert Koch. I have sequenced the name of this scientist and the work they have performed in a very lucid and a simplified manner. This is very useful. People will find it very useful. And so next, after Lucretius and Girolamo Fracastoro, who stated that invisible living creatures cause disease, then Sir Francisco Stelliuti, he observed bees using a microscope that has been provided by Galileo. Then Robert Koch, he made a first handmade drawing on microorganisms, the fungus mucor. Sir Anthony van Leeuwenhoek, he is the pioneer. He constructed simple microscope composed of double convex lens. He developed microscope with magnification approximately 300 times. Nowadays magnifications, he at that time he made it very he made a very useful thing. The most interesting technique he developed was microscopic view of liquid specimen. So we can call that Anton van Leeuwenhoek is the pioneer of microbiology. So now, as I discussed in the previous slides, the spontaneous generation. Francisco Redi, who performed experiments to challenge the theory of spontaneous generation. His experiments were primarily on decaying meat and its ability to produce maggots spontaneously. Now I'll explain this experiment in brief. What he did, Francisco Redi, he used 3 containers with meat in it. The first container was uncovered, second covered, and the third one was mesh covered. He observed that the uncovered flask which was left, flies laid eggs on the meat and maggots developed. Obviously, if the flask is uncovered, the flies will come and will sit on the meat and they will lay their eggs, and finally the maggots will develop. The covered flask, Francisco Redi found that the meat did not produce maggots. Obviously, it was covered and flies cannot enter into the flask, so no maggots will form. And the third one, the mesh covered flask, he observed that the flies laid their eggs on the mesh. So, because the flies attracted, on the smell, because of the smell of the meat, they came on the flask but could not enter, but laid their eggs on the cover, the mesh, and the eggs produced and maggots. So these are the three things which Francisco Redi performed, and what he concluded? He concluded that maggots were produced from the eggs of the flies, not from the meat. Previously people believed that the meat, which is a food, contains some organic substances. Obviously it contains organic substances, but they believed that the meat actually produced these microorganisms. But no, Francisco Redi has disapproved and stated that it is not the meat or the organic substance which is present in the meat does not produce maggots. It is the flies which produce maggots. So spontaneous generation was first discarded by Sir Francisco Redi. Next, John Needham. He boiled mutton broth. Mutton broth? No, broth is a liquid substance. He boiled mutton broth and then tightly closed. First he boiled the broth, mutton broth, and then he closed that broth. Most of the mutton broth containing flask became cloudy and contained microorganisms. Most of the mutton broth, what happened with it? It became cloudy and contained microorganisms. But you see in the previous slide, when it was closed, no maggots were formed. Okay? But you see in the experiments of John Needham, he boiled the broth, mutton broth, and then tightly closed it, but found that after closing also, after the vessel has been closed, microorganisms still were, not microorganisms, some organisms or the media became cloudy. How it is possible? He thought organic matter on the broth conferred life on living organisms. John Needham explained that there is no point of closing uncovered, mesh covered. The organic matter on the broth conferred life on living matter. So it's a controversy. It became a controversy. He stated that yes, the spontaneous generation is right. It is the organic substance which is making this broth cloudy, and finally the maggots are produced. Later, Sir Lazzaro Spallanzani, he filled a glass flask containing water and seeds and placed in boiling water. No growth was seen as the flask remained sealed. John Needham first boiled the mutton broth and then closed. He did not close the boiled mutton broth at the time of boiling. He first boiled it and then closed. So in that duration, the microorganisms might have entered the vessel. The medium was air. The medium is air. So by air, the microorganisms might have entered the broth and contaminated that broth. But Lazzaro Spallanzani, he just developed this experiment of John Needham. He used a filled flask containing this water and seed in it, and alongside he placed in boiling water. He used a closed flask and kept on boiling, So he found that no growth was seen, and the flask the flask remained filled. As long as the flask remained no growth was seen on the flask. So he concluded that it is the air which contains germs. He boiled, and after some time he closed. By this time, in that duration, the microorganisms entered. But he used a closed flask and then boiled. So there is no chance of getting contamination because the air is blocked from using this tightly closed flask seal. So Lazzaro Spallanzani, in controversy with John Needham, concluded that it is the air which contains germ. So spontaneous generation was again disapproved. Later, Sir Theodore Swan, in his experiment, air was first passed through a red hot tube. Actually, it is a modification of Lazzaro Spallanzani's experiment. He In his experiment, air was first passed through a red hot tube and then allowed to enter a flask containing a sterile nutrient solution. The flask remains sterile. Theodor, what he did, he used the air, he allowed the air to enter the flask. He did not use any sealed container before the experiments or at the time of boiling. He allowed the air to pass through the flask, to enter the flask, but the air was passed through a red hot tube. So you can imagine, if the air can pass through a red hot tube, automatically the air which contains the microbes will die. So there is no chance of contamination, and found that the flask remains sterile. So it is the air which contains germ. It is actually a support to the Lazarus Palangelis experiment, or a more modified version of this experiment. Next, Sir George Friedrich Schroeder and Theodor von Dusch allowed air to enter a flask of heat sterilized medium after it had passed through sterile cotton wool. No growth occurred in that medium, but the air was not heated. Sir George Friedrich Schroeder, these are very important names, you must remember it. This Sir George Friedrich Schroeder and Theodor von Dusch, they allowed the air to enter the flask, and the medium which was there inside the flask, it was already heat sterilized. But this time they have not used the red hot tube to heat the air. Instead, they used a cotton wool to close the flask. So definitely any people can understand the germs will will try to enter the flask, and these things, you can understand that it is this okay, yeah. So air could not enter the flask because the sterile cotton wool was there as a stopper on the flask. So automatically the germs present in the air will get trapped in the cotton wool, and he found that no growth occurred in the medium. But the air was not heated. It was not heated, but he used a cotton wool and found that no growth was seen. Sir Felix Pauchet claimed that the microbial growth could occur without air contamination. Yes, microbial growth can occur without air contamination also. He explained. It can occur by water contamination. It is not necessary that air will contaminate this medium, the medium inside the flask. Suppose you are using a mutton broth, okay, and you have used a mutton broth and you have not you have not cooked that mutton broth for the right time. So automatically microbes have a chance to grow inside this broth. So he stated that it is not the air which can contaminate the medium inside the flask. So air contamination is not the only possibility to bring the microbes inside the flask. If you don't properly cook the food, microbes will be there inside in the broth. So he was right. Next scientist, Sir Luis Pasteur, he placed that cotton plug, the cotton plug used by Sir George Friedrich Schroeder and Theodor von Dusch. Luis Pasteur modified their experiment. He placed the used cotton plug, which was earlier used to stopper this flask. He used that cotton plug in sterile medium after the air was filtered through that plug, and microbial growth was seen. Yes, the wool, cotton wool, which is used as a stopper to close this flask, has trapped microbes in it. And if you use this used cotton wool and place it in the flask, automatically microbes will grow in the flask, because the cotton wool has already trapped these microbes. And if you place this cotton wool in the medium, microbes will definitely grow, because the microbes has been trapped in these cotton wools. He observed that the cotton plug which was used for filtration was trapped with spore-like objects that might have contaminated the sterile medium. Sir John Tyndall and Ferdinand Korn demonstrated that dust carries germs, not the air. They used a very modern technique. So they have given a modern technique. They have given a modern approach. What is the approach? It is not the air which contains germ. It is the dust which contains germ. Later we will understand. We will go with practical microbiology. If you use a HEPA, high efficiency particulate air, that is the laminar airflow, there also air is coming, but the air is filtered air. So there is no chance of contamination. We do all the microscopic experiments inside the laminar airflow. So there is no chance of contamination or any microbial growth, because the air is already filtered. But if you use a dust air, automatically the dust contains microorganisms. For example, in an operation theatre room, there is very less chances of contamination. Why? Because the air inside the operation theatre is very sterile air, purified air. So there is no chance of microbes in this OT rooms. They use a high efficiency particulate air. They pass the air through filters. After passing this air through filters, they circulate this air in the room. So there is no chance. So the air does not contain microorganisms. It is the dust which is present in the air contains the microorganisms, not the air. So a sterile air would definitely not contaminate medium. So from there, many things are developed. Now later, if you read this slide twice or thrice, you will automatically understand how the microbiology has evolved. The scientists which they have experimented from there, they have developed so many mediums for the growth of microorganisms. And Sir John Tyndall's and Ferdinand Korn's experiment is helpful to make a sterile air. Okay, thank you. Next slide. So, our next slide will discuss about the microorganisms and diseases. Sir Agostino Bassi. He was the first to confirm that microorganisms cause disease. It is not by any supernatural existence that cause disease. It is the microorganisms that cause disease. He found silkworm disease was caused by a fungal infection. Silkworm to make silks, silk threads, and they found that there is some problem in finding the good quality silk. So what may be the reason? So he found that there might be some fungal or some fungus which is creating problem to this silk, contaminating the silk, and finally these silks are not getting the best quality. Productivity is not that much because of this fungal infection. Sir M.J. Barclay, he found great potato blight disease was caused by a water mold, which is, this disease is caused by a water mold. Sir Heinrich D. Barry showed that smut and rust fungi caused cereal crop disease. He suggested a method for heating the wines to destroy the undesirable microbes. The process is called pasteurization. It is like heating wines for longer times so that there is no chance of microbial growth. You see, you heat milk, right? You heat milk. This is a process known as pasteurization. Because if you don't heat milk, the milk will get spoiled. The spoiled is not caused by any supernatural god or existence. But people that time believe that it might have been caused by supernatural existence. But no, this is the bacteria which is causing this disease, milk to get spoiled. So Sir Heinrich did various experiments about pasteurization, like heating the wines may kill these microbes. Okay. Sir Joseph Lister developed a system of antiseptic. He was a surgeon that time, so he developed a system of antiseptic surgery to prevent microorganisms from entering wounds. Before that, there was no system of antiseptic surgery. People just cut the wound, cut the area where the dissection needs to be done in an open air. But after this experiments, Sir Joseph Lister found that there is a very, this is very important this time to do antiseptic surgery. Means doing the surgery in a sterile, uncontaminated room, so that no microorganisms can enter the wound. Okay. Now I'll talk about the Koch's postulate. Sir Robert Koch. He demonstrated the role of bacteria in causing disease from his study and experiments on anthrax. He found that there is a direct relationship between microorganisms and disease. Oh yeah, Robert Koch. Now he has accumulated many experiments and finally concluded, yes, it is the microorganisms which is responsible for disease. He also explained it is the bacteria that cause disease. The anthrax that time was a very deadly disease. That time anthrax is caused by a bacteria. Okay, so these are the Koch's postulate applied to tuberculosis. These are mentioned as it is because we cannot change the postulates. We cannot make it give these postulates by our own writing. So these Koch's postulates are here listed. So I'll explain it. You can read it. The microorganisms must be present in every cases of the disease, but absent from healthy organisms. He stated that for every case of disease, it is a microorganisms. But if you see a healthy organism, there is no microbes found in their body. The suspected microorganisms, if you suspect, this is the disease that has been caused by a microorganism, your first task is to isolate the microorganisms and then grow that microorganisms in a pure culture. You have to take this microorganisms from a diseased body or a person who is affected by the disease, and you need to isolate and grow in a pure culture, to grow the microbes in that pure culture. The same disease must result when the isolated microorganisms is inoculated into a healthy host. After the growth of the microorganisms in the pure culture, if you use these microbes and inoculate in a healthy organism, the same disease will occur. The same and after the disease has been occurred, if you take the microbes from that diseased body or the affected body, the same microorganisms must be isolated, which was isolated before also. The same microorganisms you will find from the diseased host. Host is the body where the microorganisms has affected or have taken, but the microorganisms which have grown in that body. This is the host body. Thank you.